Okay. Hi, I first wanted to thank the organizers for giving me this amazing opportunity to present some of our preliminary data expressing functional DRESS within the non-human primate. And although we're not quite ready to show you any behavior, and I hope none of you stand up and leave after you find out that I'm not going to show any behavior, I wanted to emphasize the groundwork that we had to put in order to get these DRESS functioning within non-human primates. And so first I wanted to start off with what our lab is interested in and why we felt that it was important to sort of be able to manipulate the brain circuitry within the non-human primate. And so what we're interested in is action selection, and so how an animal can perform the same action, such as a lever press or a dowel pull, under two different action strategies. One that could lead to goal-directed behaviors, so actions that will occur and change dependent on what the outcome of that action is, to those that are habitual. And these are actions that are thought to be more stable, so they'll occur regardless of any changes in the outcome. And so we know that cortical basal ganglion circuitry is involved in these two different action strategies. And so the area of interest for us has been the dorsal striatum, which in this rodent picture is green. And it receives converging inputs from all area of the cortex, as well as glutamatergic input from the thalamus. It receives inputs from the substantia nigra, dopamine inputs, serotonergic inputs from the dorsal raphe nucleus, as well as GABAergic and cholinergic inputs from local interneurons. And so the cool thing about the dorsal striatum is that it can be divided up into subdivisions based on its cortical inputs, as well as their proposed roles in these action selection type behaviors. And so, for example, the dorsal medial striatum in the rodents or the caudate in primates receives inputs from these higher cortical areas, the associative cortices, like the orbitofrontal cortex and the prefrontal cortical areas. And they're thought to be involved in these goal-directed or action outcome type behaviors. On the other hand, the dorsolateral striatum in the rodents, or its equivalent in the primate, the putamen, receives inputs from these sensor-motor cortices in the motor cortex, premotor area, and they're thought to be involved in these habit formations. And so we're really interested in understanding how an animal can flexibly switch between these two different action strategies in normal behaviors, as well as how aberrant habits can be formed in states such as stress, like I am right now, or addiction is what we're particularly interested in. And so using rodent models of alcohol addiction, we've sort of honed in on the dorsolateral striatum as an area that has been shown to be highly altered as an animal increases their alcohol drinking, so longer periods of time. And we also found that in postmortem tissues of monkeys that have been chronically imbibing for over a year, that these same circuits are affected. And so in order to be able to translate this to the human, we wanted to ask our hypothesis of whether this brain region here, the dorsolateral striatum or the putamen, can be seen as a target for therapeutic interventions. And so, so this is why we wanted to go into the non-human primate. One, they drink similarly to humans. They can drink different levels. You don't really have to force them to drink. They kind of like it. And so they have similar brain circuitry to humans. And so we think that this is sort of a really important area of interest. And so we asked a really basic question. Is there a technique that we can use that can reversibly manipulate brain circuitry to probe its involvement in these complex non-human primate behaviors? And so we had three criteria that we wanted a technique to meet for us to use. And so the first is that we wanted it to be reversible. So anyone that does behavior knows that there's a large variability from individual to individual when it comes to behavior. And so we wanted a technique that we can examine within the same animal what their normal behaviors are like and how that is manipulated with whatever it is that we're trying to screw up along the way. And so the second thing is we wanted to have this technique to be able to target one cell population versus others. And lastly, we wanted to decrease invasiveness. And I know this kind of sounds a little bit weird, but what we wanted to do is we wanted to be able to examine these non-human primates performing behaviors in sort of an open field sort of way. We didn't want to have to instrument them in any way. We didn't want to have to implant cannulas. We wanted them to be able to roam within their cages and sort of move about without having to head fix them. Not saying that's a bad thing, but that's sort of what we wanted to do. And so, this is not a new problem, and there's lots of different techniques that have been used to manipulate brain circuitry. And so first there are lesions. And so lesions, once you kill off those brain areas, you can't sort of shock them back into life. And so it's a non-reversible. So we didn't like that technique. And then there's GABA A receptor agonists and electrical stimulation, both which can be reversed. So once the GABA A receptor agonist sort of washes off, you'll return back to baseline. And when you turn off your electrical stimulator, you also return back to baseline. However, there's no cell type specificity or no cell type specificity with either of these models, so we decided against that. And so next we decided to move into viral expression, which will allow for cell type specificity based on the promoter that you use in order to drive the expression of non-native receptors. And so there are different options that we could look at. One was the Drosophila elatostatin receptor, which is a GI-coupled protein receptor. And so the con for this receptor is that the ligand that is used to activate it doesn't cross the blood-brain barrier. And so that would sort of, it asks for the need to have to implant a cannula within the brain. So we didn't really want to go with that. And then there's optogenetics, which I think is a very powerful tool. It allows for temporal resolution. And so you can actually ask specific questions of whether or not certain stimulus patterns is important for given behaviors. And so I think this is a great tool. However, in my eyes, I really wanted to stay away from having to have an animal tethered while they're performing a given behavior. And so that led us to looking at designer receptors that are exclusively activated by designer drugs, or DREADs. And so DREADs come in a lot of different flavors now. There are now muscarinic receptor DREADs, there's kappa opioid receptor DREADs. But when we started these experiments, we sort of focused on these two muscarinic receptor DREADs. The HM3DQ, which is a GQ-coupled receptor that ultimately activation of this receptor will lead to neuronal excitation. We also looked at the HM4DI DREAD, which is a GI-coupled receptor. And so activation of these receptors will lead to neuronal silencing. And so the cool thing about these DREADs is that they've been mutated in such a way that they no longer have an affinity for their native ligand, acetylcholine, but they've gained an affinity for clozapine and its inert metabolite, clozapine N oxide. And so clozapine N oxide is a great tool. It's a great ligand for it, and that it's highly bioavailable in both rodents and humans, and it's pharmacologically inert. So if you just apply CNO to a mouse, you don't really see any change in behavior in the absence of DREADs. And in rodents, it's a great tool because it doesn't actually back metabolize. However, it's been shown to back metabolize into clozapine in guinea pigs and humans. So the first question we ask is, if we apply different doses of clozapine N oxide peripherally to the non-human primate, how much of that actually back metabolizes into clozapine that we can measure in both the brain and the CSF? And does this lead to any off-target effects that could sort of affect any of our behavioral paradigms? And so we did a pharmacokinetic study where we injected CNO. We administered 3 different ways: IM, IV, and orally. And then we collect blood and CSF at different time points after drug administration. And so comparing the 3 different routes, we see that administration IV leads to the largest concentration of CNO in both the blood and the CSF. And it actually obviously gets into the blood, into the brain a lot quicker than the other methods. When we look at clozapine, although the concentration of clozapine in the IV is higher than the other forms, the concentration of it within the brain isn't that high compared to the oral administration. And in fact, the only animal that we saw an off-target effect was in this animal that sort of drove this error right here. And with that animal, we saw changes in heart rate, and it didn't look too good. The vets weren't too happy with me at that point. So anyway, we feel that we found an appropriate route of administration for the CNO. We like the IV method, and we can understand the timing of the event. So we know that we can start to look at behavior 1 hour after administration of this drug. And so next we wanted to get the dreads inside the brain. And so for that we use MRI-guided stereotactic surgeries. And so this slide is just to remind us that primates are very different than mice. So when we do mice surgeries, oftentimes I just look at my Allen Brain Atlas and find the stereotactic surgeries, the coordinates that I need to do in order to get to certain brain regions. However, in monkeys, you can see that both of these monkeys are the same age, although they're different sexes, but you can see that the coordinates are quite different. And so there's a high individual variability from monkey to monkey. And so the importance of getting MRIs to guide our coordinates is very important. And so after we get our coordinates, the animals are taken to surgery where we perform a craniotomy and the viruses are injected directly into our brain area of interest, the putamen. And so we've looked at 4 different viral vectors so far. All of our viral vectors use an AAV1 serotype, which has been shown to have great expression within the striatum, great spread. We looked at 2 different promoter sequences. One is a human synapsin gene, which allows for targeting to neurons. And then a CAM kinase 2 alpha, which within the striatum leads to preferential expression within projection neurons, so the medium spiny neurons of the striatum. And all of our vectors have an mCherry tag, which allows us to later on look for the viral spread as well as functional assessment of these dread receptors. So 30 days after injection of these viruses into the brain, the animals were sacrificed and we collected the brains for immunohistochemistry. So this is just an example of a monkey that was given 1 injection of 50 microliters of the HSyn-GQ dread into the putamen. And you can see that there's expression within the putamen. And if we kind of zoom in, we can see that there's individual neurons that are expressing mCherry. And if we look at the downstream areas of putamen, or the areas that the medium spiny neurons project to, we can see that there's fibers within the GP and the substantia nigra. And also, not shown here, but if you look at the cortical areas that project to the putamen, we don't see any staining. So all of this is an anterograde expression from these viral vectors. And so once again, I said that we could use mCherry to sort of assess whether these receptors are functional. And so we could take sections of the putamen. This is the putamen here. This is the callosum and the IC. And we can search for red fluorescent neurons, and we could target them for patch clamp electrophysiology. And so using, under the current clamp configuration, we can sort of test or examine what these cells, the action potential firing of these cells before and after CNO. So medium spiny neurons are pretty quiescent. They don't actually fire when they're at their resting membrane potential. And so here you don't really see much activity here. But when we bath apply CNO, very slowly in this slide, did it finish? You get an increase in firing in the putamen of cells that were injected with a GQ dread. And so, giving you examples of some of the effects that we've seen, what we find is that these are mCherry cells that were expressed with a HM3 dread, and we find that what the CNO actually does is it depolarizes the membrane. So the action potential characteristics aren't changed. We don't see any changes in action potential threshold, the half-width, or amplitude. But what we're seeing is a change in the membrane potential. So from here, it goes from -80 to -48. And on wash-off, it kind of goes back down. And you can see that here. This depolarization leads to an increase in firing of this medium spiny neuron. Now, recordings from non-mCherry expressing neurons that are within the same slice leads to no effective CNO. And so in the putamen of monkeys that were injected with a GI dread, we find the opposite to be true. So application of CNO leads to a hyperpolarization of the membrane. So in this case, it's -68 to -74. And so we could see that this here, the hyperpolarization of the membrane, that is also recovered after washout. And so we know that We know that we can get the dreads within the putamen, and we know that we can that they're functionally active. And so there are things that we sort of learned from our experiments. And one is that there is a need to perform a dose response within individual monkeys. We can see from that PK study, you can see that there was quite a bit of variance in our studies, or quite a bit of error, which suggests that individual monkeys might metabolize CNO differently from each other. And so getting individual dose response seems to be very important to be able to know how much CNO is getting into the brain and to ensure that you're not getting enough back metabolism of clozapine to cause an off-target effect. Next, we wanted to be able to obviously test this in behavior. And lastly, we wanted to figure out non-invasive measures to see if these dreads are actually working in the monkey. So in the data that I showed you before, we had to sacrifice the monkey in order to make sure that the dreads were there and to see if they were functional. But with the help of Chris Cronky and Kathy Grant at the Primate Center, we came up with this idea that perhaps we can use fMRIs to measure dread activation within the brain. And so we hypothesized that that using the T2 weighted signal, that application of CNO to monkeys that have been, or cells that have been infected with an HM3-DQ dread, application of CNO will lead to an increase in neural activity, whereas the opposite would be true in cells that were expressing the DI dread, or the brain regions that were infected with the DI dread. And so we did perform this experiment. We injected an HM4-DI dread directly into the putamen. And so we used as a seed, the putamen as well as the cortex that projects to the putamen. And what we find is that an injection of CNO IV while the animal is in the scanner, the cortex seems to chug right along. We don't see a difference in the cortical, the signal. However, in the putamen, we see a drastic decrease in the signal. And so we think that this is a great way to ensure that the dreads are functional within the brain, but perhaps two, we can actually use this as a way to get a dose response for CNO. And so maybe we can actually modulate how much activity we're suppressing or increasing using this method. And so thank you.
Thank you, Dr. Carson, for that great talk. We have some time for questions. There's still a couple of mics on either side over there.
All right.
Oh, yeah. There's a couple questions, of course, they're down here. Yeah.
I almost got away.
It's just a lot to process.
Oh, just a quick question. Have you thought of using the fMRI to look at what other parts of the brain then might be affected when you either activate or silence the putamen?
Yeah, so we are thinking of looking at fMRI to see if any of those circuits are changed, see if any connections are strengthened or weakened by our CNO administration.
This one over there.
So I have a quick question. Was there anything, was there any downsides to these particular AAVs that you used that you noticed? Was there any reactive astrocytosis or gliosis that was unusual with these?
We didn't actually look. You could see that the expression was quite patchy, and we're not actually sure why it's patchy. In all of the staining that we've done, we've noticed this sort of patchy expression, and whether that's due to inflammation or not, we haven't actually looked at yet.
Beth?
Just a quick question. So could you also use the fMRI to get a sense of your expression? So without having to go to histology, do you think you could in the live monkey actually get a sense of whether you got patchy expression or whether you're off target?
So I don't think that the fMRI will allow for that sort of resolution in order to be able to see how far out we've spread or where the patches are directly within the tissue, and I think that still needs to be done with histology. But it will sort of tell us if we hit our target or not. So, because we're picking the putamen as our seed, because that's our target region, we know that if we increase the CNO and we don't see a response, perhaps we had missed our target. But we think that performing the MRIs before surgery will really get us to, the appropriate coordinates that we need in order to not miss our target. And the putamen's fairly large, so it's kind of hard to miss.
Okay, are there any other questions? Great.
Thank you.
Thank you so much.